Oh, thank you, Edmund. That was lovely. And thank you all for inviting me. This is a lovely conference in a fascinating city, so I'm really happy to be here. So I'm giving 2 talks in this conference. The talk about the regulatory aspects comes tomorrow, not now. This is going to be a little bit of a different talk. I'm going to be talking about assays. But first, disclaimers. I have no disclaimers for the first time in 30 years. I have no disclaimers to talk about. And but I do want to say that the data you will see here was actually generated by what was my lab at the FDA. So it is their work that I'm citing. Is it working? It's not working. These are all my slides. I hope you enjoyed them.
The one below, the button below.
This one?
Yeah, this one.
Okay. There we go. All right. So, as I said, this is not going to be a fully regulatory talk, although there will be some regulatory aspects because, I can't help myself. But what I'm going to be talking about mostly is the assays that have to do with the assessment of innate immune activation as part of the immunogenicity risk assessment. And I'm going to be talking about, what the existing assays are, some gaps. But most of all, I'm going to be describing a new method that we developed in the lab to look at the impact of innate immune modulating impurities or innate immune modulating products in a more detailed way. So the classical thought when looking at immunogenicity risk assessment has always been that it is extremely hard to predict. It's hard to predict which patients are going to, how many patients are going to develop it, which patients are going to develop it, and what the clinical consequences are going to be. This is because immunogenicity is not just the result of the product, but it's a result of the product plus the environment, plus the patient, plus the disease, plus probably a few things that we're not totally aware of. So all of those interacting results in something that has been classically hard to predict, although we're getting better at it. The expectation is that because it is, as has been described this morning, really it can have such serious consequences, is that you start thinking about immunogenicity risk assessment way before you even start doing your clinical studies. That, hopefully you're thinking about it as you develop your molecule, and so that you have a fairly good understanding by the time you get into phase 1, 2, and 3, and that the methods that you develop will accompany the development of the product throughout the clinical development and post-licensing. Now, if clinical studies are the gold standard for developing immunogenicity, non-clinical studies can really help inform the risk and help mitigate it. Right? So in the regulatory context, how can you use them? I think there's been a lot of talk this morning, how do you use these to select your targets, to improve your targets, to develop better products. But what can you do with this information in the regulatory context? Well, informing, using these methods to inform the immunogenicity risk can help support the design of your clinical studies. So you can go to the agency and say, look, this is the risk, and this is how we understand the risk, and this is how we plan to assess it and mitigate it. So it can support that. It can also be used to reduce or replace clinical trials for follow-on products, such as generic peptides and oligonucleotides, support manufacturing changes, 505(b)(2), and eventually biosimilars. Now, you've heard a lot about the different assays, so I won't go into describing each one. I tend to put these assays into 4 big buckets, and I apologize if I don't want to sort of ruffle any feathers, but I put them into 4 very big categories. There's assays that are in silico, and those are looking at, trying to predict what epitopes are going to bind to MHC, or what amino acids are going to be facing the T cells. And there's a few initial studies looking at B cell epitopes. They're still quite crude. And they're not quite there yet. There's a number of assays, such as the MAPS assays, or I believe the reveal assay, that look at actual affinity, or look at what the cells are actually, APCs are actually presented capable of presenting in the context of MHC, by APCs, or by looking at affinity of the peptide or the epitope for MHC. There's a number of assays that look at what the T cells do in response to that. Do we have T cells that can respond? And then there's a last bucket that should actually be the first bucket, because it's what I really like, which is the bucket that has to do with the activation of the antigen-presenting cells. And that one is critical. Why? So we have, as I said, assays that predict what's going to be presented in the context of MHC, with what affinity, and what T cells are going to be responding. But it all really starts here. It starts with the antigen-presenting cell and the ability of that antigen-presenting cell to take up the antigen, process it, and present it. And there is a definition of immunogenicity. It's not mine. It's from Christian Nadler for about 10 years ago, that says that immunogenicity is really antigenicity in the context of an inflammatory milieu that results in a successful humoral response. And I really like that. And I think I like the definition, because I think it sort of really puts it in frame the importance of the innate immune system has in the process. And I would like to direct you to a couple of papers that came out maybe 2-3 years ago, using studies with subjects that had been vaccinated. They actually looked at 820 subjects receiving 13 different vaccines. And what they did is they looked at what was the inflammatory status at the time of vaccination and what was the titer of antibodies that they achieved. And they could really see a very nice correlation, indicating that really the immune status, the innate immune status at the time of being exposed to an antigen, really can determine or can contribute to the development of an immune response. Now, going back to Nick's talk this morning, is it worth doing this type of assays? Is it something that it's worth engaging for? There was a recent survey that was done in biotechnology and drug development companies for biologics, and about 70% of the companies are running some or all of these assays. That, I think, tells us that these are helping with development. But for this talk, again, I'm just going to concentrate on this last set. And these assays, I've sort of put them all in a big bucket, right? And you have assays that are based on PBMCs, on whole blood, on dendritic cells, on cell lines. And basically, you take your cells, you put your product in, and then you see what gives. It can be cytokines, it can be CD markers, it can be antigen uptake. What you're looking at is, are these antigen-presenting cells getting activated? In our group, we liked looking at multiple parameters: inflammation, APC activation, chemokines, stress, CD markers. And the reason why we like that is that we think that multiple parameters are better than one parameter. And I'll get into that in a minute. Now, I want to clarify for those of you who might want to go out to the FDA at any point that FDA does not recommend any one method. You can use the method you like, but you have to justify your selection. Now, all of these methods have important advantages, maybe over animal-based methods. And that is that they use human immune cells, right? You sort of, again, getting closer and closer to the patient or the human population that you're trying to reach. They're rich in pattern recognition receptors. They have that mixed cell environment that allows the cells to sort of talk to one another and amplify a response. They're generally, not incredibly complex, and can be standardized so that you can get a pretty high throughput. And they're pretty sensitive to some PRR agonists at least. They do have some disadvantages. As Oleg was mentioning, they can be quite sensitive to in vitro masking due to formulation. The other disadvantage is that we use PBMCs, or we use whole blood because it's accessible, it's easy to reach, but it may not be the best platform, right? I believe Tim was mentioning that there's low frequency of cells that are important. So if we think about it, and we look at sort of the cells of the innate immune system, in terms of antigen-presenting cells, the ones that you care about are dendritic cells and macrophages. Those are your really good antigen-presenting cells. And then, basophils, eosinophils, mast cells, very important for allergies and hypersensitivities. All of those cell types are present in very low numbers in whole blood. And what that can do is that it can result in masking of our response, as we will see. So this is another way of looking at it. If you have a whole bunch of cells and you have a few that are getting activated with stimulation, it is very hard to see the difference. But when you identify the cells of interest and then look among those cells, which ones are getting activated, that becomes a lot clearer. Now, as I was saying, we'd really like to look at multiple parameters. Why? Because we think that innate immune modulators can act on many different type of responses, elicit pro-inflammatory cytokines, but also have like anti-apoptotic effects, or regulate cell cycle. The number of things that can happen is quite varied, and they don't always happen at the same time. So there's timing issues, there's response issues. So if you do like a MIIAA assay and you just look at IL-6, you might find it or you might not. The other problem is the reverse problem. If you see one signal, it may be just stochastic. The truth is that, we see a little bit of an increase in IL-6. It's not huge, but it's there, or in IL-8 or in IL-21, and we're like, okay, there is a signal there, but what does it mean? It's kind of hard to guess. But if you see multiple markers changing, all on the same path, you can be fairly sure that you are seeing the activation of a certain immune path. Now, we talked about then that it would be very nice to understand what cells are getting activated. And we want to look at multiple responses, right? And now I'm thinking, okay, we have to do that by flow, and we have to start, to look at those little cell type groups. You need a number of different markers. For anybody who's done flow cytometry, you're starting to add those numbers of markers and antibodies and controls in your head, and it's getting pretty high, right? And you want to do multiple wells because of the well-to-well variability of when you're staying, and all of that, it starts to add up. So we were trying to, we were concerned that this would be very high cost and high variability, which would reduce the effectiveness. And so what we came up with was with the idea of using barcoding the cells. So basically, use 5 or 4 different dyes, live dyes, that are taken up by the cells before you even stimulate them. So the idea is basically you take the cells, the wells, you put your different combinations of dyes into the wells, then you put your cells in, and then you stimulate them. And after you stimulate them, you can throw them all in one big tube, put them all together, test them once, and then debarcode, reassign the cells to the individual wells, and look for activation. That was the idea. Now, an important part of that, being able to do that, was to understand whether the live barcoding would impact on the pattern of activation. And so we ran this assay. Sorry, this one seems to be a little bit out of focus. Where we took different wells, put different combinations of dyes, and then compare the activation or compare the expression of markers either in unstimulated or CpG-stimulated cells with cells that had no barcode, again, PBS or CpG oligos. We were particularly interested in understanding immune activation by oligonucleotides, and TLR9 activation is a particular concern, so that's why we focus on CpG oligos. But what you can see here is that once you deconvolute, you have all the different wells that were tested as one in tube A or tube B or C. And I think I lost my pointer. There's no more. Do you have another pointer? So, sorry.
Try using this one.
This one? Yep, that works. Thank you. Or not. Anyway, so what you can see is that if you look at the profile of tube A versus B and C, they look exactly the same. There was no impact. So then we tried looking at what the activation was in PBMCs with CpGs, low levels of CpG oligos. And when you look at PBMC, what you see is what I described before. It's hard to make a decision. These are very low levels of activation with high variability between donors. It becomes very hard. When you instead can separate the cells by cell types and focus on the cells of interest, and again, we were concerned about TLR9, so we focused on B cells and plasmacytoid dendritic cells, which are the cells that express TLR9. You can clearly see the difference in expression of markers, whether it's CCR7, CD25, CD40, CD69, and so forth. And so we could, whether it was a sort of fairly large population like the B cells or a really tiny one like the plasmacytoid dendritic cells, we could see it. And then, when you look at sort of at a population, then those differences become fairly obvious. And we could do that for CpGs, we could do it for R848, which is a TLR7/8, and so forth. So how can that help us when we're looking at products? In this example, we were looking at oligonucleotides. These are actually morpholinos, and we were interested in contrasting two different lots that had a difference in manufacturing. We wanted to understand, is there does this result in difference in impurities? And what you can see is that when you look at the response of the PBMCs, whether it's in unstimulated wells or wells that were actually received CpG in addition to the product, you can see that very clearly the CpG ones are activated versus the unactivated, and you can see the difference very clearly, whereas you can't see them with the PBMCs as a whole. And the same can be said when we did a similar experiment using Remicade in a biosimilar. So, is this an assay that you would want to run on all your samples? Absolutely not. But is it an assay that might be helpful if you're trying to investigate an issue, if you're trying to understand maybe, or if you have a specific concern? For example, we were concerned for oligonucleotides, is there a TLR9 activation? Is there a TLR9 activation of plasmacytoid dendritic cells, which might not lead to immunogenicity, but could lead to an increase, for example, in interference, making patients feel kind of lousy. So it is an assay that you can do to investigate products more thoroughly. So once again, in vitro and in silico assays, they can be helpful. They can inform on the immunogenicity risk, but it's just information. We have to look at the results in the context of the whole data set, not an isolated instance. I don't think that if we try to get sort of a little number that says, well, this is above or below a certain immunogenicity, that's not going to be so helpful. If we look at all of the assays together, we try to integrate all the information with all the clinical and all the other information that surrounds it, then we can get a fairly good assessment of risk. So lastly, IRMI assays or innate immune activation assays, they're designed not to detect immunogenicity. They're only designed to detect biological differences in response. And that can inform the risk of immunogenicity. It's very important to have that in the context, right? So demonstrating The corollary of that is that demonstrating the absence, if you have a very good assay and you demonstrate the absence of a difference, then you can be fairly certain that there is no issue of risk. Once again, these studies were done primarily by Mohamed Mangaisan, Seth Thacker, and Joy Balsamo. And I want to leave you with a parting thought, which is the absence of evidence is not the same as evidence of absence. But if you have really good assays, it comes pretty close, right? But you can only understand the results if you look at it at the product as a whole, CMC, clinical, clean farm, etc., and you integrate all that information so you can get a really good sense of what the risk is. With that, I thank you.
Yeah, hello. Thanks for the presentation. I noticed you also mentioned in your slides that FDA never mentioned or recommended a single method to detect this immunogenicity response. My question more to the siRNA modality. So do you think the type 1 interferon need to be measured in the non-clinical study? I'm asking due to, type 1 interferon have alpha and beta alpha have different subsets. And from methods perspective, it's very challenging. And due to their short half-life, it's also quite hard to capture their peak in the non-clinical study. So yeah, from regulatory perspective, do you think?
Let me see if I understand. Well, you're asking for siRNA products, whether you should monitor for interferon induction as a potential for side effect.
Yes, type 1 interferon.
Well, that's something that you're going to have to consider depending on what your product is. But it is, I have to say that for oligonucleotides, the FDA policy right now is not set. They have, there's guidances for about 17 or 18 products right now. And when you get to the immunogenicity part, it says, please talk to the agency. And the reason it says, please talk to the agency is because they're still learning. They're still learning, and there is not a consensus yet on what's going to be needed. So at this point, propose a strategy, give a good justification, and it will be considered. That would be my advice. But I absolutely understand what you're saying, that looking at message may be a lot easier than looking for protein. Protein just disappears. It's very hard to capture. I agree.
Okay. And my second question, I didn't get your barcoding system. How does it work? So it's the dye conjugate to different CD antibodies, or so you showed your barcoding system. I didn't get how does it work.
So the idea is that in each well, you put a different barcode. That combination of live dye markers is taken up by the cells. It's a live dye. It's taken up and the cells retain it, right? So then you can pull all the cells into one tube, run them all, forget about assay well-to-well variability. You run them all, you stain them all together, you run them all together, but then because the cells have this barcode, you can put them back, sort of reassign them digitally into the well that they came from. And then you know that particular cell, where it came from. But because you have all these different markers, you can identify all the different cells in it and say, okay, I want to look at the B cells from this treatment. They were subjected to the same treatment or staining as the other cells, and you can make more easy comparisons.
Okay, thank you.
Thank you. Thank you very much. We have time for one more question. All right, if not, thank you, Daniela Verthelyi. Thank you. Right. Thank you so much indeed. Fantastic talk. We're introducing our next speaker.